Okay, picking back up with the cytoskeleton, just like it sounds, skeleton, there, skeleton, that is the structure or the skeletal framework of our cell. So it'll tell you what type of shape you'll have with that muscle cell. It's being very elongated, or with just a skin cell. They could be cuboidal, they could be flat like an egg. So it'll determine what they're going to be shaped like. Gives it strength and allows it to move. And then we'll look at these different types of extensions that the cytoskeleton can give rise to. Well, let's go back and look at them. I don't have a slide on it. Okay, so microvilli, very tiny projections. They're shorter. But there's a lot of them. This is going to be where you need to absorb something. So you're going to see microvilli in, say, your intestines. That's a good place to look at microvilli. I don't know if you'll follow me all the way over here. Nope, it's limited. But microvilli, you're going to see little projections. And again, think about inside your intestines. This is microvilli. All throughout your intestines. And what happens, instead of food coming across just one flat tube, it can go up over this one, down this one, up and over. And if we were to stretch it out, then we've got a longer tube. So it actually, microvilli increases surface area. So that means more food can run over those little projections. That's what microvilli is for, to increase surface area. Your cilia is needed to move things. So especially right now with cold season coming up, your cilia is going to be working overtime. And so it's constantly beating, kind of beating, and it goes in one direction. So from the lungs up, it's beating up. So when you're sitting here and you're coughing up stuff, that's cilia making it come up. Or if it's in the upper respiratory tract, it'll beat down. And so as you're sitting here, you're constantly swallowing mucus. It's going down in the stomach. Yeah. And sometimes you don't even realize it. It's just slide. That's why it's constantly sliding back behind in the back of the throat. It's cilia that's making it do that. Because we want to get it out. It's trapping stuff and we want to get it out. Flagella. In the human body, we only find flagella in one place and one place only. Where is that? Yeah. So females, we don't have flagella. That's the only place you'll see in the human body that we have flagella, is in the sperm. And of course, that's for movement. Okay. Let's look at our different types of cellular transport. You're going to have to know these at each and every single one of them for the exam for sure. And again, these will not go away. We will talk about these in other systems down the road, here on out, all the way down to the very last reproductive system. So passive transport, the difference between passive transport and active transport is passive doesn't require any energy whatsoever. So we don't have ATP with passive. Active you do. So looking at diffusion, did we talk about each and every? Yes. So looking at diffusion, what happens is we're looking at particles. So these little bitty dots we're looking at. We're not worried about the water. So don't concentrate or focus on the water. In a minute we'll focus on water and its movement. So diffusion, they put a dye tablet in and then it diffuses. So these little particles start out highly concentrated here. They want to go from an area of high concentration to an area of low concentration. So this is high where this is low. Meaning there's a whole bunch of particles, we're all bumping into each other, It's uncomfortable. I'd much rather be way over here where there's nobody else. And so that's what it does. Until we all spread out and we reach an equilibrium. Another good example I like is a fart. Can start way over there by Miss Gwen, and those are actually fecal particles. So when it hits your nose and you actually smell it, it's actual feces that is hitting your nose.
That's I remember learning that in 7th grade. So now, like, every time, like, my brothers will, like, fart or something, I'm like, just tell me so I don't, like, smell it.
So I'm not, like, taking your poop inside my body.
Yeah.
Yes. So it'll start out, the particles will start out way over there in the corner, and then they'll diffuse, work their way everywhere else. And that's why it spreads.
Like your broccoli did this morning?
Just like the broccoli. They are particles that are, again, highly concentrated in the microwave, and they diffuse all throughout, even into the hallway.
And my science teacher last year, he told us that, like, if you, like, get a whiff of it every now and then, it's not in the air anymore, like, it's still in you, like, that's what you're smelling. Was he just messing with me?
No, it really does.
That's
it does stick
disgusting.
with it. Does.
Like, when we smell that hallway, that broccoli, we're going to go.
Yeah, it's in your nose. It ain't going away. Sorry, I'm moving here again.
Diffusion is a natural movement of particles from an area of higher concentration to an area of lower concentration. Diffusion is a form of passive transport. That is, it does not require energy to occur. Diffusion occurs in air or water. It can perhaps be best illustrated by placing a dye tablet in water. As the dye tablet dissolves, the particles move away from the tablet where concentration is high to the edges of the container where the concentration of particles is low. Diffusion continues until the particles are evenly distributed. The point at which no further diffusion occurs is called equilibrium. Even when a membrane stands in the way, the particles will still diffuse through it as long as the membrane is permeable to those particles. A difference in concentration of a substance from one point to another is called a concentration gradient. When the particles move from an area of greater to lesser concentration, as occurs in diffusion, the particles are said to move down the concentration gradient.
That will be important talking about a concentration gradient when we get to active transport. Okay, so we looked at particles. If we're talking about particles moving from one area to another, that is called diffusion. If we're talking about osmosis, we're talking not particles. But you still have to look at the particles to figure out which way water is going to move. So water does the same thing. It has a concentration gradient that it likes to go against. So you're looking at this side here. We've got an equal amount of water in A and B. But the thing is there more or less water in B compared to A? There is less. And the reason is because there is more dots taking up space and squeezing out water. Just like when you're sitting in the bathtub, looks like there's a lot of water to get out. So you were taking up a lot of space, you get out and the water goes way down. Whereas another bathtub right beside you could be the same level, but there's actually less water. You take your body out of it and it drops way down. So lots of particles mean less water. So where does water want to be? Where there's less or more? Less, yeah, just like the particles. So you have a hydrostatic pressure here that's higher. There's more water. They gave an example in your book about a water balloon. You know, you blow it up with water, put tons of water in it, and you fill it. That's hydrostatic pressure. There's a pressure to it. So it wants to be pushed out. So it'll go from here to here. So this is what it'll look like. It's the same amount of water. It just looks different. It's still the same. Let's look at... These take forever to load.
Osmosis is a type of diffusion that often happens when a particular substance cannot cross a membrane. The membrane in the container shown is separating a 5% albumin solution from a 10% albumin solution. The membrane is permeable to water, but not to albumin. In this situation, the water, not the particles, moves in an effort to equalize the concentration. This is an example of osmosis. Diffusion of water down the concentration gradient through a selectively permeable membrane. The concentration of the two solutions eventually equalizes, but in the process, one side ends up with a greater volume of water.
Okay, still talking about water, there is what's called tonicity. So that means more or less, if you think of tone, you know, how much strength or how much particles we have. So isotonic concentration, so an isotonic solution, you have the same amount of particles inside of water compared to the cells. So that's like normal saline. That's what we use, what we would give you in the hospital in a bag is normally normal saline. Because it's the same concentration, it's the same tonicity as our cells. Okay. We give other types of solutions, which we do in the hospital. A hypertonic solution, do you think that solution would have a lot of particles or little particles? Hyper. Lot. So, there's things that change. So, with a isotonic solution, let me get over here where my board's not messed up. In an isotonic solution, same amount of particles in the solution as there are in the cell. So I've got 1, 2, 3, 4 particles in the solution, 1, 2, 3, 4 particles in the cell. Nothing changes with the cell. So we don't have fluid moving in or out. With a hypertonic solution, oops, I want to get this out of the way. With a hypertonic solution, we have, you said hyper, we have more particles in the water compared to our cell. So, which way do you think fluid will move? Inside the cell or out of the cell? Because how much water do I have out here compared to in there? Yeah. It'll move out of the cell. Because there's a whole bunch of particles out here and only a few in there. That means there's a whole lot of water in comparison in here than out here. So water's going to want to move from a high pressure to a low pressure, so it'll move out. And that cell will shrink. It's in a hypertonic solution. So we have osmosis occurring. So in a hypotonic solution, will you have a lot of particles in the solution or few? Few, because it's hypo compared to our cell. So, will fluid move in or will fluid move out of that cell? It'll move in because there's a lot of water. A little bit of water. So there's one water molecule where there's like tons of water molecules out here. And it doesn't want to be out here. Tons of water molecules are going to move in. What's going to happen to that cell? It's going to swell. It may even lyse, which is bust, rendering that cell unusable. So we're going to look at the same thing in a video illustration as soon as it loads, 2 hours later.
Cells are essentially closed containers. Besides containing fluid, they also contain a variety of solutes such as salts, sugars, acids, and bases. It is the concentration of these solutes in the fluid that determines whether and how much fluid moves into or out of a cell. If a plasma membrane is impermeable to a particular solute, and if the solute is more concentrated on one side of the membrane than the other, osmosis will occur. The three terms used to describe these solutions are isotonic, hypotonic, and hypertonic. A hypertonic solution contains a higher concentration of solutes compared with the fluid within the cell. If a red blood cell is immersed in a hypertonic solution, such as a concentrated salt solution, water will diffuse out of the cell, causing it to shrivel and perhaps die. A hypotonic solution contains a low...
So what happens when you put salt on a slug? Yeah, because you're putting it in a hypertonic solution. You're putting a high concentration of salt particles on the outside of it, and it's got the fluid, the nasty, slimy stuff, acts as a solvent, and so then it just takes all of the moisture inside that slug out. Yeah, it would be a terrible way to die. Ugh. So that's what's happening with our cells when we put it in a hypertonic solution, just like a slug.
Die. A hypotonic solution contains a lower concentration of solutes compared with the fluid within the cell. If a red blood cell is placed in a hypotonic solution, such as distilled water, water will move by osmosis into the cell. This influx of water will cause the cell to swell and, eventually, to burst. An isotonic solution is one in which the concentration of solutes is the same as it is in the cell. When a red blood cell is placed in an isotonic solution, water moves into and out of the cell at an equal rate. As a result, the cells remain normal in size and water content. Isotonic fluids hydrate the body without causing dramatic fluid shifts.
Fluid shifts. So that's why we use isotonic pretty regularly in the hospital. We don't have any changing of fluids drastically. You very rarely see a hypotonic solution. Highly contraindicated with brain injuries. Brain injuries, you already have swelling of the brain, and then you give a hypotonic solution, what's going to happen to that brain cells, those tissues? They're going to swell up too because you're giving them a hypotonic solution. Fluid's going to move into those cells. Stop. Okay, so another passive transport. So we talked about diffusion, movement of solutes or particles. Diffusion going from an area of high concentration to an area of low concentration. Still passive. Talked about osmosis, which is water moving from a high concentration to a low concentration, or hydrostatic pressure. And now we'll talk about filtration. Filtration encompasses both of those things. Particles are going to move, or solutes, as well as water. And the reason is because of pressure. So we have a lot of pressure coming down through in this blood vessel, pushing through the little pores, the little holes in our vessels. So this is a cell, an epithelial cell, an epithelial cell. And then in between there, we have pushing through, we have particles and water because of pressure. So blood pressure is what's causing this. You'll see filtration in our capillaries. So that's where we give nutrients like glucose and oxygen, and we'll also pick up stuff. And then you'll see it in your kidneys, filtering things out. A good example in your book was about the coffee. You know, when you're making coffee in the morning, you have a permeable filter in there and you have coffee grounds, the water that's coming from the top into the filter where the grounds are is creating a pressure. Just from the weight, the gravity of the water hitting it. That's pressure. So, very tiny particles come through along with water. And the tiny particles is what gives you your coffee its color or your tea its color. So that's filtration happening. Whereas the large particles, the coffee grounds, are not moving through. So here, your large particles, your blood cells, some proteins are not moving through those holes. So that's why we said filtration occurs in kidneys. Should you see blood in your urine? No. So these are too big to be going through there. So if you see blood in your urine, then you may have a problem with the kidneys itself. Now, urinary tract infections, that's a different story. You got inflammation, and that's why you're seeing blood.
Why do they say if you get in the kidney?
Because what are you doing to those tiny capillaries that are inside a kidney? What happens when you get punched in the eye? It swells up. What else does happen? It turns black. Why does it turn black?
Because the blood cells are busting and it's...
The actual vessels that were there were busted, and so blood comes out of those vessels. You get a bruise.
Isn't that what
Same
a bruise
thing
is?
happens with, it's all a bruise is busting of the blood vessels. So you get punched in the kidney, you bruise the kidney, you've actually busted blood vessels in the kidney, and yes, you will see blood in the urine. Because it should be closed tight, no red blood cells coming out. But if we bust it open, red blood cells are going to come out, and we'll see it in the urine.
In filtration, water and dissolved particles are forced across a membrane from an area of higher to lower pressure. One of the most obvious examples of filtration in the body occurs in capillaries. The pressure of blood inside the capillaries forces water and dissolved materials, such as nutrients, into the surrounding tissue fluid. It is through filtration that the body's cells receive the nutrients they need to survive. Filtration is also the method the kidneys use to remove waste products from the blood.
So, of course, in the urinary system, we will talk about filtration once again. The last passive transport is called facilitated diffusion. Seems like this would be active, but we're not using ATP to do this. So what happens is we have these transport proteins or channel proteins that are sitting on that bilipid cell membrane, and so they're open. And there's certain things, like a solute, this could be glucose, that will fit in there. It's like a lock and key. Only one thing will fit in there. If something else rolls by and dumps in there and it doesn't fit, the channel protein will not do anything. So what happens is once this side closes, the other end opens, and it comes inside the cell. That's how glucose works, getting it inside the cell. Later on in the endocrine system, we'll talk about how insulin plays a role in all this, because insulin does help as well. The video, which isn't loading now, just illustrates that again. Okay, so this one is getting into active transport.